Welcome to Chapter 15, Special Senses, the last topic that is covered in Anatomy 1. So let's get started. What are our special senses? Well, vision, taste, smell, hearing, and equilibrium or balance. We use different receptors in each of our special senses to help us know what is going on around the world, in the world around us, where our body is in space. We use all of these to help us figure out where to go next and what to do. All of these receptors, they're located in your head. Since all your special senses are located in your head, it would make sense. So let's start with your eyes and vision. So 70% of the receptors that we have in our body, our entire body, are in the eye. And even half of that cerebral cortex is involved in visual processing. So whatever you see, your brain processes it so you know what you're actually looking at. That is half of your your cerebrum. That's a lot. We only see about 1/6 of our eye. The rest of it is behind in that orbit. It is surrounded by fat because we need some kind of cushion to protect it. And then we have that bony orbit that's around it to protect it even further. We have several different accessory structures. We have eyebrows. Our eyebrows and your eyelashes help to shade the sun. They also help to kind of block and protect your eye from any kind of anything coming at you. We have our eyelid. We have at the corner of our eye, we have what are called commissures. The one further from the center, of course, is going to be the lateral commissure. And the one closer to your nose, which is closer to the middle, is that medial commissure. We have a lacrimal caruncle, which is this right in here, that lateral caruncle. The sclera, that is the white. The palpebrae fissure, that is the opening where we see your eye. We have an iris, which is that colored portion. We have that lower eyelid. We have pupil. The pupil is just an opening for light to shine into the posterior portion of your eye. We'll talk about that today. That iris, that pupil, even part of that sclera is covered by conjunctiva. So here's a bunch of these accessory structures that I mentioned on the slide before. So I said those eyebrows, they're going to shade from light. They're also going to prevent sweat from reaching your eyes. So it helps to protect eyelids or palpebrae. They are separated by a palpebrae fissure, which is that opening. They meet in the corners to make that medial and lateral commissures that I pointed out. We have a lacrimal caruncle. It's at that medial commissure, and it has oil and sweat glands. We have what are called tarsal plates. or connective tissue, and they anchor orbicularis oculi and levator palpebrae superioris muscles. These are different muscles that help do different things within your eye. You blink your eyes every 3 to 7 seconds. It's usually better that somebody's, if you ever really wanted to count it to see how long, is they'd have to do it without you knowing, because as soon as you start knowing, you start holding your eyes open longer, you start blinking more. When you're consciously aware of something, it doesn't work as well. So we blink to protect from foreign objects, but we also blink because we have secretions that are coming out from certain glands, and we need to spread those across the eye. Because we need to moisten our eye. Our eyeballs need to stay moistened so they can help us see well. Your eyelashes are just like any other hair in the body. They have a follicle and they're innervated. The nerve endings are what allow you to blink. We have tarsal glands. Tarsal glands are sebaceous glands. They're going to produce an oil, and that's what lubricates the eye. We have ciliary glands, and these are sweat glands. So let's look a little more. You see this tarsal plate up here? That helps to hold certain muscles in place, as you can see. We also have that palpebrae fissure, which is the opening of the eye. You see this conjunctival sac? That helps to keep things from getting lost behind the eyeball. If you've ever had something get lost in your eye before, like a contact or something, it usually gets stuck up or below in this conjunctival sac, so it doesn't go into the posterior portion of your eye. If you've ever worn contacts before, you probably know what I'm talking about. What is that conjunctiva exactly? It's a mucous membrane, and it helps to produce mucus secretions that help to keep your eyeball lubricated. We have a palpebrae conjunctiva. That is on the underside of the eyelids. Bulbar conjunctiva. That's what covers the whites of your eye. The bulbar conjunctiva contains blood vessels. So if you see somebody with quote unquote bloodshot eyes, red eyes, this is when these small blood vessels in the bulbar conjunctiva actually dilate and more blood is coming to that area. And that is what causes that bloodshot eye appearance. We have that conjunctival sac. It's between the palpebrae and bulbar conjunctiva. And that conjunctival sac is actually where contact lenses sit. So there's that bulbar conjunctiva, that conjunctival sac down here, palpebrae conjunctiva, all helping to keep our eyes safe. We have what is called a lacrimal apparatus. It consists of the lacrimal gland which is at that top. So this is a portion of the eye that we see here. Here's your nose just for reference. So the lacrimal gland sits up in the lateral portion of the eye. It releases its solution out onto the eye, which are called lacrimal secretions or tears. It's kind of like a saline solution. It's got antibodies in it. It's got antibacterial lysosomes to help try to protect your eye from any kind of infection or foreign invader. As we blink, it's going to take the secretions that come out, and as we blink, it gets spread over the eye. And it's kind of heads in this direction. We have a lacrimal canaliculi and a lacrimal puncta. Here's much better drawing than what I do. So we have this lacrimal gland up here. This lacrimal gland secretes down onto the eye. The tears, which go, as we blink, it goes across the eye. It goes into the lacrimal canaliculi, which are these little tubes that go up. Now, they didn't draw these in. So these little tubes that take the secretions, take it down to that lacrimal sac, which drains into the nasal cavity. That's why when you cry, that lacrimal gland gets stimulated to secrete more and more secretions, and only it starts draining down into your nose. And as more tears get drained down into your nose, the more your nose runs when you cry. So let's look at the muscles of the eye. We have 6 muscles specifically that attach to the exterior portion of the eye. They allow you to look in several different directions. We have 4 rectus muscles, as they're called. And they come from this tendinous ring in the back. We have a lateral rectus, superior rectus, inferior rectus, and the one other muscle that we cannot see in this picture is that medial rectus. So there's a superior, inferior, medial, and a lateral rectus. These 4 muscles allow you, when the superior rectus contracts, lets you look up. When the inferior rectus contracts, it lets you look down. When your medial rectus contracts, it lets you look at your nose. When your lateral rectus contracts, it lets you look out. We also have 2 oblique muscles. We have a superior and an inferior oblique. And it allows you to kind of rotate your eyeball, move it in different directions besides up, down, left, or right, medial or lateral. So here is that superior oblique muscle. As you can see, the superior oblique muscle goes through a tendon and it goes down and attaches to the eyeball. The inferior oblique muscle, it's going to come in and go across the eyeball to help you look different directions. So here's another view. You can see this common tendon ring. You see lateral rectus, medial rectus, inferior rectus, superior rectus, superior oblique, And in this one, we cannot really see inferior oblique well. But here, we can see inferior oblique from this view. And then you can see each of the rectus muscles are exactly medial, lateral, superior, inferior. And then this was superior oblique coming through, goes through this little tendinous ring and then attaches down to the eyeball. So here is what the these different nerves do. Which directions they allow you to look, and then what cranial nerves specifically go to them. Because we talked about cranial nerves when we talked about the peripheral nervous system. So lateral rectus has abducens or cranial nerve 6 going to it. Superior oblique has trochlear or cranial nerve 4 going to it. The other 4 muscles, medial rectus, superior rectus, inferior rectus, and inferior oblique, they all have oculomotor or cranial nerve 3 going to it. So if you know that lateral rectus has abducens, superior oblique has trochlear, then the rest is oculomotor, which makes it a little easier to memorize than, okay, medial rectus oculomotor, inferior rectus oculomotor. Just lateral rectus abducens, superior oblique trochlear, then the rest of them are oculomotor. That's all you have to remember. So there's some different conditions that involve some of these glands or ducts or even layers of the eye. We have what is called a chalazion. That is when one of those tarsal glands gets obstructed and it turns into a pretty painful bump on the eye. We have a stye. A stye is when one of those sebaceous glands gets inflamed. And that's right at the base of an eyelash. It gets very painful. Conjunctivitis. It's when that conjunctiva gets inflamed. We have that itis. That means inflammation. It's when your eyes get red and they get kind of irritated. Pink eye. Pink eye is an infection that's caused by bacteria or viruses. Pink eye is highly contagious. If you've never had it before, you're lucky, but pink eye is highly contagious. And since your nasal cavity is kind of continuous with your eye because of that lacrimal system that drains down into your nasal cavity, colds or nasal infections actually can swell up that entire nasal lacrimal area. And that's why when you get sick, you get more watery eyes because you cannot drain those tears that's lubricating your eyes. You can't drain those down into your nasal cavity. So instead, they just come out as watery eyes. You've ever heard of double vision? Well, the medical term for double vision is diplopia. Di means 2. So what happens when those eye muscles that move your eyeball, they don't work in perfect unison. They're out of sync with each other. So you have a really hard time focusing on something. A lot of times someone ends up closing one eye so they can focus on it. Because your brain can usually take the visual fields from both eyes, blend them together so you only see one kind of picture. But when those eye muscles are not working well, you may see two of that same thing because you're seeing what the right eye sees and what the left eye sees. We'll go into exactly how that works, how your eyes look at different things here shortly. A strabismus or cross eye. It's usually congenital and it's a weakness of those external eye muscles. So your eye ends up kind of rotating medially or laterally when at rest because it cannot focus, or that one muscle is weaker than the rest. So that's where your eyes switch back and forth looking at one specific object. And then your brain kind of sends out information incorrectly to what you're actually seeing. So let's look at the eyeball some more. We looked at just the outside. So let's look at what all is contained within this eye. We have a fibrous layer, a vascular layer, and an inner layer. And then we have internal cavities, which are called humors. And your lens is what separates the anterior and the posterior segments of the eye. So here is your eye. That outer layer is that sclera. The middle layer is that choroid. And then the inner layer is the retina. And we said this lens, this right here, is what separates the anterior chamber from the posterior chamber. So that outer layer is avascular, so there's no blood supply to it, connective tissue. We have that sclera that we looked at up here. That helps to protect and keep the shape of your eyeball. It's where those muscles on the exterior of your eye that help you move your eye, that's where those are attached to, is that sclera. And at the back portion of our eyeball on the sclera, that's where our optic nerve that goes back to our brain to help us process what we are looking at. That's where it exits. And that sclera is actually continuous with the dura mater of the brain. Next, we also have the cornea. The cornea is the transparent portion at the front of the eye. So this part right through here is the cornea. And that is where light enters, and it also bends the light as it enters your eye. We'll talk about the importance of this later. We have epithelium, of course, across the top, that outer surface, because we need a way to protect your eye from abrasions of any kind. We have corneal epithelium, and that inner layer that has sodium pumps. Those sodium pumps help to keep that cornea clear. We have pain receptors on that cornea. So if you've ever scratched your cornea before, you would know. It's painful. So that vascular layer, that middle layer, it has the choroid, the ciliary body, and the iris. So that choroid is at the posterior portion. It has blood that supplies all three layers. It's a brown pigment. So it has lots of melanocytes that secrete melanin. That is what gives it that brown pigment. But it absorbs any light that may have scattered different places, so that the light can kind of focus into one area of our eyeball. That ciliary body, the next portion. That choroid becomes a ciliary body. And that is what is the tissue that surrounds the lens. We have ciliary muscles, ciliary processes, and ciliary zonules that all connect to the lens. The zonules hold the lens in place. The ciliary muscles are what's used to change the shape of the lens so you can focus on things at different distances. We have ciliary processes that help secrete fluid for the anterior segment of the eyeball that makes the aqueous humor. Your iris. As I said in the picture above, that iris is the colored portion of your eye. It's between the cornea and the lens. That iris has an opening in it that is called the pupil. And that is where light enters your eyeball. For close vision and bright light, those ciliary muscles will contract and that pupil constricts, gets smaller. It's that parasympathetic control we were talking about in the autonomic nervous system. For distant vision and dim light, we have this dilator pupillae muscle that does exactly that. It dilates the pupil, which is sympathetic control. Your pupils can change size during different emotional states, or different problem solving skills. Your pupils change size as you're focusing on something. So here's that ciliary body. We have that ciliary zonule, which are these little fibers through here. We have the iris, the pupil, with that lens in the middle. So here is a human eye. And you can kind of see, you see that ciliary zonule, or suspensory ligaments are the other name for it. The ciliary processes and the ciliary body. So here it shows parasympathetic control. Parasympathetic control constricts the pupil, makes it smaller. Sympathetic control dilates the pupil or makes it larger. These two muscles are located within the iris, the colored portion of the eye. Now let's go to the inner layer. That very inner layer is known as the retina. The retina is kind of an outpocketing of the brain. It has photoreceptors, millions of them. That takes light energy and lets you know what you're looking at. There's lots of neurons. glial cells. We have an outer pigmented layer and an inner neural layer. So the pigmented layer, it's a single cell thick, and it comes anteriorly and covers the ciliary body and even the iris. It absorbs light, tries to prevent scattering. It phagocytizes photoreceptors and cell fragments. And your eye is a place that stores vitamin A. So here, this entire layer through here is the retina. We have our different parts of the retina. We have the neural layer and the pigmented layer that help to, these all work together to help us see. So within that neural layer, we have different types of neurons. We have photoreceptors, bipolar cells, and ganglion cells. Information goes from the photoreceptors to bipolar cells to ganglion cells. Then the ganglion cells are what exits the eye as the optic nerve. The site where the optic nerve leaves the eye is called the optic disc. In that optic disc, it is known as the blind spot because there are no photoreceptors. The type of photoreceptors we have are rods and cones. So here's that layer, different parts of the eyeball. Again, let's look. Rods. Your rods are what allow you to see at night. They see in dim light. They're also what allow you to see peripherally when your eyeballs are looking forward. You see so far to the side when you're looking forward. Your rods do that. We don't necessarily see color very well. There's no color vision in our rods. And we don't get very sharp images in our rods either. We have the most rods in our periphery, so the outside portion of our eye. Cones. I like to think cones start with C. They do. Color vision, which starts with a C. Cones, color. Same letter they start with, so they helps you just kind of remember which ones see color, so then you just know the other ones do the opposite. They're what function in bright light. We have this portion of our eyeball called the macula luta, and it's mostly cones in that area. The fovea centralis is where we have our best vision, our best visual acuity, and it's all cones. The fovea centralis, our eyeball likes to move around when we're trying to focus on something, because our eyeball is trying to focus that image directly into that fovea centralis so we can get the best view of what we're looking at. So here is the different layers. And there's the pathway of light. Light's coming in and hitting this way. It kind of goes through to these rods and cones first. The rods pick up the dim light, cones pick up the color. Those then will go to these bipolar cells. Some of them will go to what are called horizontal cells through here, but they'll go then to these ganglion cells. We have some amacrine cells throughout that kind of also help us with our different vision and like integrating different pictures together. But then they come from these ganglion cells, they come out here and they form that optic nerve. So here is a pictomicrograph of our eye. You can see the rods and cones, the bipolar cells, and the ganglion cells, and in between are usually the axons between each. So our eye gets blood different ways, two different ways. The choroid, that choroid layer supplies the outer third or the photoreceptors. Then we have a central artery and a central vein. The central artery and central vein enter and leave the eyeball with the optic nerve. It's within that optic nerve, actually. And we can actually see these blood vessels when we look into the eye. See? This is looking directly into the eye using one of those otoscopes. Ophthalmoscope, sorry. Otoscopes go in the ear. Ophthalmoscope goes through the eye. This is using an ophthalmoscope to look into somebody's eye. Here is that optic disc. All these blood vessels are from the central artery and the central vein. Here is that macula luta with the fovea centralis, where your most accurate vision is, and it is all cones at this portion. A retinal detachment. Your retina can detach from the posterior portion of your eye. And the back posterior portion of our eye is filled with vitreous humor. And what will happen is it's this vitreous humor that's like a jelly. We'll see it when we dissect cow's eyes in class. But this vitreous humor gets between the retina and that choroid layer, and it can lead to permanent blindness. I had a teacher in school describe it as closing a curtain. He said he's had it. It closes a curtain. Some of those blackout curtains that gets rid of all light, but it just kind of goes black where you don't see anything. Your retina can get torn doing different traumatic activities. Bungee jumping, because it's a sudden stopping of head movement. They can reattach it with laser surgery. So at least it is fixable. So here's that anterior and that posterior portion of the eye. Again, the lens, the ciliary zonules are what separates that anterior and that posterior segment. So in that posterior segment, we have that vitreous humor, which is kind of like a jelly. It helps to get light onto that retina, but it's also supposed to help hold that retina against that choroid, that next layer down. The vitreous humor helps to keep your intraocular eye pressure because it helps to hold that shape of that eye. It forms when we're an embryo, and this vitreous humor can literally last our whole lifetime. The anterior segment is filled with aqueous humor. We have two portions to the anterior segment. We have an anterior chamber and a posterior chamber. The anterior chambers between the cornea and the iris, posterior chambers between the iris and the lens. The aqueous humor that fills both the anterior and posterior chambers of that eye is constantly being recycled and remade. We have blood vessels, capillaries, that go to those ciliary processes that I mentioned above, slides before. Well, those capillaries are filtering out plasma that gets used for aqueous humor. It's a plasma-like fluid. And it drains in the scleral venous sinus back into the blood supply. So that aqueous humor in the front is being made and drained, made and drained, made and drained constantly. How we get nutrients and oxygen to that lens and that cornea, even the retina. But that's also how we get rid of wastes in our eye, because yes, we do get those. So here is that ciliary process and that ciliary body that secretes that aqueous humor. That aqueous humor fills this whole portion. It comes through and out and fills this entire anterior chamber before it drains at this scleral venous sinus, here and here. Glaucoma. Glaucoma is another condition that has to do with the eye, but this time it has to do with the fluid in the eye, specifically that aqueous humor in the front portion of the eye. We're not able to drain that aqueous humor, so we get too much aqueous humor in our eye. Having too much aqueous humor increases the pressure in that eye. And it can get so high that it can compress the retina and the optic nerve and eventually cause blindness. There's not many early signs for glaucoma. We don't notice it usually until later when we start seeing halos around lights. You start seeing you get blurred vision. But that's why after you turn 40, you should be getting a glaucoma test done yearly to make sure that your aqueous humor is being drained properly. So that treatment is eye drops. Those eye drops help increase that drainage of aqueous humor, or it can decrease the production of aqueous humor. You can do laser therapy or you can have surgery done on it as well to help fix the glaucoma. Lens. Lens is a biconcave disc that can change shapes to focus something onto that retina. We have fibers that help to make up this lens that are called crystalline. And these fibers are constantly being added. So the lens, as we get older, becomes more dense, convex, and it loses that elasticity as we age, so you can't focus things as well. So as we get older, our lens can start to cloud. It could be just a consequence of aging. It happens to some people. I've seen it happen to several dogs as they've gotten older. Diabetes can cause it. Heavy smoking. Frequent exposure to intense sunlight can cloud your lens. Sometimes it's congenital, but that crystalline protein, it starts to clump, which is what causes that clouding, is it's more clumping of that protein instead of fibrous. Vitamin C increases cataract formation, which is that clouding. We can use artificial lenses to replace a clouding or a cataract lens. So this is what a clouded up lens looks like. Can't see very well into the back of the eye. You see all the different clumps throughout. So how do we focus? Well, our eyes focus based off of wavelength and color. We have electromagnetic radiation, means that all energy has waves. The longest waves are radio waves and the shortest are X-rays. Here is visible light. It is very small portion of all the different rays. Our eyes can only respond to this visible light. So what is light? Light is packets of energy or photons or quanta. They travel in a light always travels in a wave-like fashion of some sort. Different waves, different wavelengths depending on the color. So red wavelengths are the longest and have the lowest amount of energy, while violet are the shortest and have the most energy. Our eyes, the color that we perceive is based off of that wavelength. Grass is green because it absorbs all colors except green. This is some physics concepts. White reflects all colors, while black absorbs all colors. That's why when you wear black clothing outside on a hot, sunny day, it's absorbing every light ray, which makes it warmer. If you wear white outside on a hot summer day, it's cooler because all those light rays are being reflected off. So here's those different wavelengths. We have cones in our eyes specific for blue, green, and red. We have those specific cones that pick up those specific colors, and then they pick up different mixtures of colors to get the rest of the different colors of the rainbow. Refraction is the bending of light rays. And that happens because as the light goes through some sort of transparent medium, it's going to change its angle. Like if you've ever watched water with light shining through between water and air, the light bends. Well, your lens does a lot of the same thing. Your lens can refract light because it is curved on both sides. But our lens can also, it is normally convex, but it can go concave. So when a lens is convex, it'll bend the light going through it So they come together, all the light rays that are coming in to this. See, these are my light rays coming in at all these different directions. They go through that lens and they come out as one piece. So they converge. All those light rays converge at a focal point. When the image comes in, it's upside down and reversed. So this is what we normally see. This is what our retina sees. Upside down and reversed. A concave lens prevents light from being focused. So as the light rays come in, they're gonna scatter more. So they use just a picture drawing with colors to show how the different colors, see the purple in the bottom corner down here goes to the top corner, orange going from the bottom corner to the top corner, purple from this top corner, light purple, then yellow, how they all switch places and move around. That's showing how the light is switched, what comes in versus what we interpret, what our retina sees, and then our brain switches it back. Our brain's pretty cool. So what is that pathway of light entering? Well, first it's got to hit that cornea. That cornea is the first place that light bends. Then it goes to the aqueous humor to the lens that'll bend light some more. Once it goes through the lens, it goes to that vitreous humor. Then it's going to go to that neural layer of the retina to the photoreceptors, which are going to finally send off information. Light is refracted 3 times. The cornea, when it enters the lens, and when it leaves the lens. Most of the refractory power is at the cornea, though. But that cornea cannot change the focus. That is where that lens comes into play. The lens can change its shape for distant vision versus close vision. So you see that ciliary zonules are these little fibers. These little fibers are what are used to help change the shape of that lens to focus in on stuff that's close up versus far away. Our eyes do best at distant vision specifically. So distant vision is where you're far away from that point, usually about 20 feet for emmetropic eyes. Emmetropic means normal vision, where you have that 20/20 vision. And that cornea and that lens, they work best at focusing something from 20 feet away directly onto the retina. At that point, those ciliary muscles, those zonular fibers, are relaxed. So that lenses are stretched flat at this point. So this is a 20/20 vision here. Light comes in, you can see how as it hits the retina, it is flipped and reversed. But the lens kind of flattens out for distance vision to help us see more clearly. What about for close vision? Close is looking at something from under 6 meters away. So our eye has to accommodate. And that is where our lens comes into play. Our lens will change the shape to increase the refraction so we can still get that image onto that cornea where it needs so we can see what we're supposed to see. Near point vision is the closest your eyes can focus. The younger you are, the closer you are to where your eyes can still focus on whatever you're looking at. The older you get, the further something needs to be away from your face before it can focus. Presbyopia. Welcome to aging. It's where you cannot accommodate. It's usually over the age of 50. That is where people start needing readers type of glasses so they can see things up close because their lens cannot accommodate things up close anymore. We talked about this a little bit in the autonomic nervous system, but we have that accommodation pupillary reflex where the pupil changes its shape. So the accommodation pupillary reflex constricts the pupil and it prevents divergent light rays from entering that eye. That's your parasympathetic nervous system. Convergence is that medial rotation of the eyeball because you're trying to focus in on something. Your medial rectus muscles allow that because your medial rectus muscles pull your eyeballs so they can look at something between the eyes. So here's where close vision. That was parasympathetic fibers. They contract. That's where these wavy lines are coming in to show that these zonual fibers are loosened up. That lens bulges up more so we can see something that's closer to us. So we have different types of vision. That emmetropic eye we said was normal. We have what is called myopia or nearsightedness. So here is this normal eye. Light rays come in and they focus in just fine. Myopic eye. The eyeball is too long. So the point where your eyeball goes to focus something, there's nothing there. That is where we need concave lenses to correct it. Hyperopia. The eyeball is too short. That is with wearing corrected lenses. So here is hyperopic. That focal point is behind that eyeball because the eyeball is too short. They need a convex lens to fix it so they can see clearly. The last type is an astigmatism. We have unequal curvatures of the cornea or the lens. They use a cylindrically ground lens, or they can use laser procedures to fix it. We do have an astigmatism chart in the lab. People with an astigmatism, here's the kind of cool part about it. They can see either horizontal or vertical lines clearly with their astigmatism eye, but everything else around that is a blur. Kind of fun to play with. I have an astigmatism in my right eye and I can only see vertically clear. Everything else around it is a complete blur. So let's look at phototransduction. So our photoreceptors, our rods and our cones, they're modified neurons. They have a cell body, they have a synaptic terminal, and then they have two different segments. Your outer segment is what receives the light. That's that visual pigment. They have an inner segment that joins that cell body, and it's connected by the cilium to the outer segment to the cell body via outer fibers. The cell bodies of these photoreceptors, they're connected to the synaptic terminal via inner fibers. That plasma membrane, the outer fibers folds back to form discs. So these photoreceptors can get damaged very easily. They can degenerate. They can be destroyed by very bright light. Here's a cool thing. The outer segment of these photoreceptors are renewed every 24 hours. The tips of the fragments come off, and then they're phagocytized. So here are these different photoreceptors. They are named because of how they look. Cones, rods. See, this is a rod. This is a cone, named based off of their shape. Here is parts of these discs of this rod that are being phagocytized off because they're replaced every 24 hours. Rods are very sensitive to light because remember, they are best for night vision for dim light. So they're very sensitive to that bright light. They don't like being in that very bright light. And that's why they're in that peripheral vision and they work well at night. They let you see tones of gray only. That's why if you ever get up in the middle of the night to go to the bathroom or something, what you see is pretty fuzzy and it's not always very clear. You can thank your rods for that. Because the rods only give us kinds of gray tones, we could have 100 rods that come together to one specific ganglion for the neurons. It's kind of crazy how many rods come to one place versus cones. Cones are for bright light and color vision. They react quicker. They only have those 3 pigments that we looked at. But that's what allows you to see color so well. They do not converge. That's why we have such high resolution in color. Some cones even have their own ganglion cells, especially in that fovea centralis, because that's where our most accurate vision is. And so in that point, the cones have to have their own ganglion cell to attach to. So here is the chart from your book that talks about rods and cones, what they're for, how well they see, how many, where they're mostly located. Color blindness. If you've ever known anybody who was colorblind, it's kind of fun to play with, just to kind of see what colors they can and cannot see. They usually lack one or more cone pigments. Could be red, could be blue, could be green. And it's usually inherited as an X-linked condition, which means it's more common in males. Since males only have one X chromosome, females have two. 8 to 10% of males have some form of color blindness. The most common is red-green. So either red cones are absent or green cones are absent, one or the other. So red can appear green or green can appear red, just depending on which cone is missing. Retinol. That is a light absorbing molecule that's going to combine with some different proteins, usually opsin, to help form visual pigments. Opsin comes, or retinol specifically comes from vitamin A. And 4 opsins are rhodopsin, found in rods, and then green, blue, and red cones. Cone wavelengths kind of overlap a little bit, and that's what allows us to see many different hues of colors, because it can trigger more than one cone to help visually see something. Like yellow light stimulates red and green cones, but if there's more red, we see orange. So if you've ever played when you were little, played with mixing different colors of paint together, taking those 3 primary colors and seeing what kind of colors you can make out of it, well, that's exactly what your cones are doing in your eyes, so you can see different shades of different things. We have retinal isomers. They're what allow us to see in 3D. They're three-dimensional objects. We have 11-cis retinol, which is bent, or a straight form, which is all trans retinol. So here you can see in this rod disc, specifically, you see that retinol, which are these little red dots, and the opsin, which are the spiral parts to it. This is part of the photoreceptors. So what is transduction exactly? It is the process by which pigment captures photon light energy, then it is converted to a graded receptor potential. As in, light comes into our eye and it creates an action potential that goes back into our brain. That's phototransduction. So deep purple pigment of rods is rhodopsin. Those rods are in that outer segment. So we have different stages of rhodopsin formation and breakdown. We have pigment synthesis, pigment bleaching, and pigment regeneration. Pigment synthesis. Opsin and 11-cis retinol combine to form rhodopsin in the dark. Pigment bleaching. Rhodopsin absorbs light. 11-cis isomer of retinol changes to all trans isomer. When retinol and opsin separate, rhodopsin will break down. Then we have pigment regeneration. All trans retinol is converted back to 11-cis isomer rhodopsin is regenerated in the outer segments. So this is what's happening between light and dark. So step number 1, this 11-cis retinol here, it is derived from vitamin A. And it is combined with opsin to form rhodopsin. Pigment bleaching. So this light is absorbed by the rhodopsin, and that triggers a series of steps which changes its shape from 11-cis to all trans, and eventually it'll releases from opsin. So here's this all trans retinol. Then we have our pigment regeneration, where enzymes slowly convert all trans retinol to its 11-cis form in cells of the pigmented layer. We need ATP to make that happen. So light and dark, this has to do with light and dark adaptation, being able to see going from a dark room to a light room, or from a light room to a dark room. That's what this is about. So rhodopsin. It is very sensitive that it can cause bleaching just by looking at stars. Bleaching is kind of where you see that white light, you see white. So rhodopsin in bright light, bleaching occurs so fast that rod's virtually non-functional. So you're going from somewhere dark to somewhere bright. Your rods can't see anything. Your cones can't see anything. The cones respond to that bright light, but then if you go from that bright light to somewhere dark, that's why you kind of see a white light before things kind of fade out. So light adaptation. So when we move from darkness to bright light, we see a glare. Rods and cones are both stimulated. Pigments are broken down. We get a glare. Your pupils are going to constrict. It takes 5 to 10 minutes for that rod system to turn off and those cones to adapt. Dark adaptation. When moving from bright light into darkness, we see blackness because your cones stop functioning. Bright light has bleached those rods. So they're still turned off. It takes some time for them to fully adapt. Your pupils start to dilate to try to help you see. Rhodopsin accumulates in the dark. Retinol sensitivity starts to increase. Transducin returns to the outer segments. It takes 20 to 30 minutes to fully adapt to being in darkness. It's crazy how long that can take. Night blindness. Your rods don't function well. So if your rods can't function well, you cannot drive safely at night. Could be due to rod degeneration. Could be because of a vitamin A deficiency. Could be caused by retinitis pigmentosa, which is a degenerative retinal disease that destroys the rods. Those tips of the rods get sloughed off, but they're not replaced. So how do we process and relay visual information to our brain? Well, as information leaves the eye, it leaves via the optic nerve. We have this optic chiasm right through here. Different parts of that optic chiasm will cross over, but that optic chiasm or chiasma forms the optic tracts. Those optic tracts will go to the lateral geniculate nucleus of the thalamus. From there, they form optic radiations, which will go to the primary visual cortex and the occipital lobes, which will then go to the visual association areas around that visual cortex. Other places, those tracks will go as the superior colliculi, so we can move our eyes. Some of our ganglion cells have melanopsin or a circadian pigment. And that's going to go to the pretactile nuclei for your pupillary reflexes or the suprachiasmatic nucleus for the daily biorhythms of like your sleep-wake cycle. So here's information that's coming into the brain, or coming into the eye, I should say. As it comes into the eye, depending on where it's coming in from, you see where these yellow fibers, some of them cross over in that optic chiasm, others do not. Same with the other side. They're going to different places. They're going to that lateral geniculate nucleus. Then they're going to follow this optic radiation back to the primary visual cortex. They're also going to go to the association areas around it to help you associate what you're seeing with something you've seen before. So here is this pathway in the brain, in a real brain. It's kind of cool. You see this optic radiation through here. You can see this optic chiasm. This is part of that optic nerve that's kind of come off. So both of our eyes, of course, are going to focus in on the same thing. But we see it, what you see in your right eye versus your left eye it's the same thing, but at different angles. So because of these different angles we see things at, it allows us to see objects in 3-dimensional. Because your cortex can actually visually place that information to be able to see exactly where it was at, which leads to that depth perception, how far away it is, how large it is. But we need input from both eyes to make that happen. So what happens if we lose an eye? Or one of the optic nerves, something happens to it? Well, you're not going to be able to see peripherally on whatever side that damage is on. If something happens past that optic chiasm, then part or all of the opposite half of the visual field is lost. A stroke can affect the left visual cortex, And by that, it causes blindness in the right half of the visual field. So our retinal cells, they're going to split into channels that sends information about color, how bright something is, angle, direction, speed of movement. We have lateral inhibition, and that is because of the amacrine or the horizontal cells. These ganglions are going to pass information to that lateral geniculate nucleus of the thalamus. And that's what helps to process our depth perception. Cone input can be emphasized on that depth perception. Our primary visual cortex, it has a topographical map of the retina in it. Neurons respond to dark and bright edges. Form, color, motion, visual association areas. It's all done at that primary visual cortex. And that visual association area, because it helps you to associate it with something you already have known maybe. So all of our information will go on to the temporal, parietal, and frontal lobes, because objects can be identified and located in space. Like you see something, you want to pick it up. Well, it has to go to that motor cortex so you can actually go pick it up. Let's move on to our sense of smell. Vision is the most complicated out of all of our senses. The rest of these are pretty easy comparably. So your sense of smell or olfactory epithelium is your organ of smell. It is located in that superior portion of the nasal cavity, and it's part of that superior nasal concha. We have olfactory sensory neurons. Just like we have bipolar neurons in the eye, we have bipolar neurons in our nose. That is what helps make up our olfactory sensory neurons. We have supporting cells. Then we have olfactory stem cells. So here's this olfactory nerve. The olfactory bulb that has all of these little finger-like projections that come down. That is our nerves that help us detect smell. So our olfactory neurons here, they are bipolar. They're kind of apical dendrites that have knobs at the end. They have non-motile cilia or olfactory cilia that radiate from that knob. So they have these little ciliary projections that come down, and they're covered with mucus because smells must be dissolved into this mucus for us to be able to smell something. They are non-myelinated axons that are part of that olfactory nerve or cranial nerve 1. Olfactory nerves. They have stem cells because we get new neurons every 30 to 60 days. These are the only neurons in our entire body that regenerate constantly. Why? Because we'll completely lose our sense of smell if they don't. So every 30 to 60 days, we're getting new neuron processes coming down to help us keep our smell. So here is, I think you can see these olfactory cilia that make up the dendrites of these bipolar neurons. These are those olfactory sensory neurons. You see these little stem cells, those purple cells up here? Those are stem cells that are going to form new neurons. This is the cribriform plate of that ethmoid bone. Remember looking at that ethmoid bone when we looked at bones in the lab? Well, this is all those little holes is where these bipolar neurons come down into our olfactory epithelium to help us smell. This is that olfactory bulb we described, and then we have mitral cells, which are the cells that help to transport information from whatever we have smelled off to the brain. So a single smell can contain up to 100 different odorants. And we can smell approximately 400 different smells. It's crazy, huh? So each of these smells encodes a different receptor protein. One protein can respond to one or more odors, just depending. So each odor is going to bind to several different receptors, and each receptor has one type of receptor protein. Different irritants, like ammonia, can actually cause like a pain or a temperature type. Like chili peppers or menthol. So if you've ever smelled menthol before, it smells cold. Chili peppers smell hot. Ammonia smells painful. So those bipolar neurons in our olfactory epithelium, they're going to synapse with those mitral cells that are in that olfactory bulb, as I'd pointed out in the picture. They synapse at what's called the glomeruli. So these axons from different neurons, they converge on a glomerulus. These mitral cells can amplify, refine, and relay signals. They can release GABA to inhibit mitral cells so that only highly excitatory impulses are transmitted. Yeah, and this is where our nervous system does some pretty cool stuff. Sends some information off and other stuff that's saying, no, that's not as important. We don't need to send that off. This is what needs to go. So those mitral cells, they travel on olfactory tracts to this piriform lobe of the olfactory cortex. Of course, we're going to send information to that frontal lobe. It's going to go through the thalamus. Going to go to that hypothalamus, amygdala, and even other regions of the limbic system. Because a lot of times we get some kind of emotional response to a smell. Like home-baked cookies in the oven. That smells really good. And it kind of gives this nice pleasure type of sensation out of it. And our amygdala lets us have that pleasure type of smell. Anosmias. That is where You cannot smell well. Could be due to a head injury, inflammation in the nasal cavity, neurological disorders like Parkinson's. We can also get olfactory hallucinations. Somebody who has epilepsy and gets seizures, they usually have some kind of olfactory hallucination before a seizure begins. That's an aura. And that way once they pick up on that, they usually know when a seizure is coming. Taste. Sense of smell is pretty easy. So our taste. We have different kinds of taste buds. Actually, more than 10,000 taste buds on our tongue. They're located in what are called papillae or projections. We have fungiform papillae, volate papillae, and valate papillae. The fungiform papillae are more mushroom-shaped, and that's where most of our taste buds are, is the fungiform papillae has most of our taste buds. It's all over our tongue. The volate papillae are on the sides of the tongue. The valate papillae, those are the largest. They have 8 to 10 that form a V at the back of the tongue. We have some on our soft palate. We have some on our cheeks, even our pharynx and our epiglottis. So here is our taste buds. You can see the fungiform, the volate, and even those here that are those valate papillae. Which makes that V shape on the posterior portion of our tongue. So each of our taste buds, they have these flask-shaped epithelial cells. We have gustatory epithelial cells that have these little hairs on them that create taste pores that's bathed in saliva. Again, we use saliva to help us taste. Within these gustatory epithelial cells, we have sensory dendrites. And they send signals to the brain based off of different tastes. We have three different types of gustatory epithelial cells. One that releases serotonin, one that lacks synaptic vesicles, and one releases ATP as a neurotransmitter. We have basal epithelial cells. These are those stem cells. Basal is usually going to mean some sort of stem cell, just like in the skin, and they're dividing every 7 to 10 days. So here's this valate papilla. Here's all these little taste buds, these little purple portion things in here. Those are those taste buds that connects to a nerve that goes down, that'll send information to our brain. Here are these gustatory hair cells. You can see all the yellow fibers going in. Those are the nerves. These are these little gustatory hair cells that help to bring the tastes. They're bathed in saliva, and they bring the tastes in and they go into the nerves. We have 5 tastes: sweet, sour, salty, bitter, umami. Sweet, like sugar, saccharins, alcohol, even some amino acids. Some lead salts taste kind of sweet. Sour. Hydrogen ions. Salty. Sodium chloride. That's table salt. Bitter. Different alkaloids like quinine and nicotine. Caffeine is bitter. Aspirin. If you've ever chewed aspirin, it's bitter. Umami. Glutamate, aspartate, like beef or cheese. Monosodium glutamate. There's thoughts that there's possible sixth taste out there, where we can taste long chain fatty acids from lipids. And I think that's why people like fatty foods, because we can taste that and it tastes good. So different tastes, what we like and what we don't like, they have different homeostatic values because they were trying to help us protect us. Like certain bitter things specifically, a lot of people kind of have that ew gross to bitter things because it's a protective mechanism. Because a lot of poisons are bitter and we're trying to protect ourselves. Even sour. There's something that could be spoiled. To be able to taste some kind of chemical, we have to be able to dissolve it in our saliva. It has to diffuse into a taste pore and contact those gustatory hair cells before it can go on to the brain so we can determine what it is. We have 3 different cranial nerves, 2 main and 1 other cranial nerve that does most of our taste. Facial nerve, cranial nerve 7, so the anterior 2/3 of the tongue. The glossopharyngeal nerve, the posterior half, and the pharynx, that vagus nerve that does everything. Epiglottis, lower pharynx. So as we're swallowing stuff down, we can still taste it. Taste, that's going to trigger reflexes for digestion, like the secretion of saliva into our mouth to help dissolve food, but also the gastric juices in the stomach. By us tasting stuff, it secretes more gastric juices in our stomach to help break up the food even further. Taste can also initiate a gag reflex or reflexive vomiting. Other protective mechanisms. 80% of your taste is through smell. So if you ever had a cold and you can't really taste anything, if your nose is stuffed up, blocked, everything tastes bland. Your mouth has thermoreceptors, so it can detect hot versus cold. Mechanoreceptors for texture. Nociceptors, like hot chili peppers. Temperature and texture enhances or detracts from taste. Like some people can't eat certain textures of foods. Some people really enjoy spicy hot foods. It triggers pain receptors, but some people experience it as pleasure when eating hot, spicy foods. Taste disorders aren't as common as smell disorders, just because taste receptors have 3 different nerves, those 3 cranial nerves that go to it. That's partly what helps save our taste. Because there's very few reasons that all 3 of those cranial nerves would be damaged at the same time. So we could have a taste disorder because of an upper respiratory tract disorder or infection. We just can't taste anything. It just tastes bland. Head injuries. Well, head injuries can do a lot of things. Being on certain medications or chemicals. Well, of course, that's going to affect your ability to taste. Head and neck radiation for cancer treatment. It is shown that somebody taking a zinc supplement can help with radiation-induced taste disorders. Cool stuff, the cool research studies that are out there. Hearing and balance. You notice smelling and taste, they're pretty simple. So let's move on to our ear. We have three parts to our ear. We have the external or the outer ear, the middle ear, and the inner ear. The external and the middle ear is where we hear. The internal or inner ear, we have hearing and our equilibrium or balance at that portion. We have different receptors for hearing and balance. So that external ear, that auricle or that pinna, that is your ear, that whole cartilaginous part of your ear. It acts as a funnel to funnel sound into your ear. That helix is the rim. The lobule is that ear lobe. We have an external acoustic meatus, which is that auditory canal. It's got some hairs in it just to help kind of protect it from things getting lodged in there. Sebaceous glands. Ceruminous glands that produce earwax, again, to help prevent things from getting stuck in your, getting too far into your ear, causing damage. We also have eardrums, or that tympanic membrane. It is between the external and the middle ear. It's translucent. It's made of connective tissue. And this tympanic membrane is going to vibrate depending on sound waves. Just like we talked about light waves, now we're going to talk about sound waves. And what this does is it transfers sound to the bones that make up the middle ear. So here is this auricle or this pinna, the helix, the lobule that all make up the ear. It works as a funnel to funnel sound waves into that external acoustic meatus. It's going to come up on this tympanic membrane. This tympanic membrane, when it hears sounds, like if you're hearing me talk right now, is going to vibrate to these sounds. And as this tympanic membrane vibrates, it's going to go to these bones that make up the inner ear. Or middle ear, sorry. Well, this middle ear, it's a small air-filled cavity. It's in that temporal bone. On the opposite side of that tympanic membrane, opposite wall to it has an oval window and a round window. We have an epitympanic recess in this area, which is the roof of the middle ear. And we have the mastoid antrum, which is where those mastoid air cells in the mastoid process are. So let's look. So here's this tympanic membrane. This portion all through here is what makes up that middle ear. We have the round window here, we have the oval window here, and we have these three little tiny bones, the malleus, incus, and the stapes. Malleus because it looks like a hammer, incus is like an anvil, And stapes is like a stirrup. That's how they got their name. We also have this pharyngotympanic or auditory tube. That is what connects the middle ear to the nasopharynx, or the back of the throat, nasal area. Used to be called the eustachian tube. So if you hear somebody say that, it's the same thing as the pharyngotympanic auditory tube. It's usually kind of flattened out, but if you yawn or swallow, it can be used to equalize the pressure in your ear. So if you've ever been on an airplane before, as you go up to different heights or levels into the sky, you have to equalize out the pressure because you're arriving at different pressures in the air as you fly higher and higher. So you're popping your ears or yawning or swallowing to equalize out that pressure to match the external air pressure outside on the other side of that tympanic membrane. If both sides are not equal pressure, that tympanic membrane doesn't vibrate and you can't hear as well. Sounds start to get distorted at this point. So those three bones that I pointed out in the middle ear, they're known as the auditory ossicles. We have the malleus, incus, and the stapes. They said they're named for their shape. Malleus the hammer, incus the anvil, stapes the stirrup. They're all synovial joints because one articulates with the next, which articulates with the next. That stapes is what articulates with the oval window. We have different muscles. We have a tensor tympani and a stapedius. They help reflexively contract to loud sounds so we don't damage our hearing receptors. So here, that tensor tympani and the stapedius muscle. These two muscles help kind of muffle out the sound. So when you're like at a concert, a really loud concert, so we don't damage our hearing for being one night at a loud concert. Otitis media. It's a middle ear infection. A lot of kids get a middle ear infection with a sore throat because as a kid, that pharyngotympanic tube is more horizontal. It's also shorter. And it is the most frequent cause of hearing loss in children. It can cause the eardrum to bulge outward and become inflamed. Antibiotics, most of the time, will help relieve the infection. We have in our inner ear a labyrinth. Again, it's in that temporal bone and it's behind our eye socket. We have the bony labyrinth and the membranous labyrinth. This bony labyrinth is a system of channels and cavities that kind of worm through this bone. We have the vestibule, the semicircular canals, and the cochlea. The bony labyrinth has perilymph, which is very similar to cerebrospinal fluid, while the membranous labyrinth has endolymph. But this membranous labyrinth has membranous sacs and ducts in this bony labyrinth. So let's look. Here is this cochlear duct in the cochlea, which is this whole circular thing. We have our different semicircular canals. They're at different directions, and we'll kind of talk about why. We have a utricle, a saccule. Then as I show those semicircular canals, we have our different nerves that come into the area. So let's go into this some more. We have our vestibule. This vestibule is found through this area in here. It contains that saccule and that utricle. That saccule is part of that cochlear duct. The utricle is part of those semicircular canals. They have equilibrium receptors or balance. They respond to gravity, changes in position of the head. So as you're looking left to right, up or down, they're responding. So you know where you are in space at all times. Within these semicircular canals, they are, as I pointed out in the picture above, in three different planes. We have an anterior, a lateral, and a posterior canal. The anterior and the posterior are located at right angles to each other. A right angle is an angle that is at 90 degrees. We have a lateral canal that is horizontal. We have semicircular ducts that line the canals and communicate with this utricle. We have an ampulla. That is where our equilibrium receptors that are called the crista ampullaris are located. These receptors, they kind of respond, as I said, to angular rotational head movement, so looking left and right. They help you in the ampulla do this and keep your head steady, and it doesn't cause you to get dizzy by doing this. So here's this utricle and that saccule. That utricle is bigger and it is continuous with these semicircular canals. You have the crista ampullaris and the ampulla. Then you see that anterior canal, the posterior canal. They said they were about 90-degree angles from each other. Well, here's that 90-degree angle right there. And then we have that lateral that comes around. So let's look at the cochlea. The cochlea is this whole spiral portion through here. It is about the size of a pea. That's really tiny. And it extends off of that vestibule. It has a cochlear duct that has a spiral organ, or the organ of Corti is what it's called. And it ends at the cochlear apex. It is divided off into three separate chambers: the scala vestibule, the scala tympani, and we have another portion of the scala tympani throughout. That picture didn't show it, I was just checking. The scala vestibule goes to that oval window that has perilymph. The scala tympani has endolymph. And the other part of the scala tympani ends at the round window and has more perilymph in it. The scala tympani and vestibule, they're continuous with each other. They have at their apex the helicotrema. We have a vestibular membrane, which is the roof of that cochlear duct that separates all this out. We have a stria vascularis. That's what secretes our endolymph. That floor, we have a bony spiral lamina and a basilar membrane. And in this spiral organ, we have cochlear hair cells. They are arranged in rows depending on height. We have inner hair cells and outer hair cells. And they all go to this cochlear branch of the vestibulocochlear nerve of cranial nerve 8. So here we kind of see through this. We have the helicotrema, which is at that apex. We have the cochlear duct, the vestibular membrane, the osseous spiral lamina. We have the spiral ganglion. This is that cochlear nerve of the cochlear part of the vestibulocochlear nerve of cranial nerve 8. So here you see the scala vestibule that has perilymph, scala tympani that has perilymph, the scala media, this whole part through the middle here, this is endolymph. And within this, we have that spiral organ. Here is that spiral organ. You see these hair cells. We have the outer hair cells and the inner hair cells. These inner and outer hair cells connect to this tectoral membrane. So as this tectoral membrane moves, it stimulates these hair cells. And as these hair cells, those hairs start to move, is what helps to generate the action potential that goes down these cochlear nerves into that forms that vestibulocochlear nerve, cranial nerve 8. So here are those hair cells in this electron micrograph. We have the inner hair cells at the top, while the outer hair cells at the bottom. We can see how these hair cells can move, and as they move, they're going to generate action potentials. So we have those semicircular canals. Inside the semicircular canals, we have semicircular ducts. They're going to help with equilibrium. Some of them help with acceleration, linear acceleration, like driving in a car. Putting your foot on the gas really fast causes you to go forward fast. That's linear acceleration. And that is done at the vestibule, where the utricle and the saccule are. They're also going to be able to tell you where your head is in space. So it sends all that information into your brain so you can consciously know where your head is in space. We have this cochlea that has that cochlear duct. That is where hearing occurs. So let's look at exactly how hearing happens. So these hearing receptors respond to sound waves. So a sound is coming in to our ears. It's going to cause that tympanic membrane to vibrate. As this vibrates, we have those three bones, malleus, incus, and stapes, that attach. The vibration of this tympanic membrane causes the malleus, the incus, and the stapes to start vibrating. As they vibrate, it causes this oval window here to vibrate. And as that oval window vibrates, it's going to stimulate the cochlear hair cells to send impulses to the brain for interpretation. So sound is technically a pressure disturbance. So when sound waves are created, they're created by objects moving. Different air molecules get displaced, and it helps to push forward to different areas. As the objects return to the normal position, we have fewer air molecules or rarefaction. So these sound waves. So we have these altering sound waves that are coming through, and this is what is getting directed into the ear by that auricle, that whole outer ear that acts as a funnel to take these sound waves into their ear, which is causing that tympanic membrane to vibrate. So as that tympanic membrane is vibrating, it's causing waves. And as these waves vibrate, it causes those ossicles inside the ear, those little muscle or those little bones, to also vibrate. And that is where kinetic energy, energy in motion, gets transferred to air molecules, which is transferred to other air molecules. And that is how we get this wave. A tuning fork vibrating creates these waves. So frequency. Frequency is the number of waves during a given time. A pure tone has repeated crests and troughs. Crests go up, troughs are the down part. Up, down, my horrible drawing. Wavelength is the distance between two crests. So from here to here, that is one wavelength. The shorter the wavelength, the higher the frequency of the sound. The higher the wavelength, the lower the frequency of the sound. Or the longer the wavelength, sorry, the lower the frequency. We can hear between 20 and 2,000 hertz or waves per second. We hear best at 1,500 to 4,000 hertz or waves per second. Pitch is the different frequencies. Higher frequency, higher pitch. The quality or the characteristic of sound. Most sounds that you hear are going to be a mixture of different frequencies. Tone is one frequency, like a tuning fork. A tuning fork only plays one sound. And that sound quality is what creates the complexity of sounds and music. So here's the different frequencies. In the red, it's a high frequency, short wavelength, high pitch. Blue is a low frequency, long wave, high or low pitch. So that amplitude or the height of the crests is perceived as how loud. Again, it's measured in decibels. Normal range is 0 to 120 decibels. Talking to somebody is around 50 decibels. Pain is about 120 decibels. Prolonged exposure over 90 decibels can cause severe hearing loss. A rock concert is usually over 120 decibels, which is where sometimes pain, you perceive pain during loud music. So high amplitude is loud, low amplitude is softer. So as I've already mentioned on that first slide, sound waves come in, they go through that external auditory meatus or auditory canal to that tympanic membrane. That tympanic membrane is going to vibrate. Higher intensity, the more vibrations. That vibrating of that tympanic membrane is going to cause your auditory ossicles, the malleus, incus, and the stapes to start vibrating. And it's going to send that vibration from that tympanic membrane or your eardrum to the oval window where the stapes attaches. The tympanic membrane is 20 times bigger than that oval window. The scala vestibule, that stapes rocks back and forth on that oval window with each vibration. And that's what causes the perilymph to move. And because the perilymph is right on the other side of that oval window. We're going to go through that helicotrema path where waves with frequencies below threshold hearing travel through the helicotrema and the scala tympani to the round window. We have the basilar membrane path where sounds in hearing range go through the cochlear duct, vibrating the basilar membrane at specific locations according to sound frequency. So sounds coming in, it goes through these three ossicles, vibrate, which causes this oval window here to vibrate. It's going to go through the scala vestibule. Depending on the wavelengths, it could go all the way to the helicotrema. Then they come back and they're going to go back out the round window. Resonance. Resonance is movement of different areas of that basilar membrane in response to particular frequencies. The basilar membrane is going to change along the length of the whole thing. So fibers that are closer to that oval window are short and stiff because they're going to hit high frequency waves. Fibers near the cochlear apex are longer and floppier because they're going to resonate with lower frequency waves. So that basilar membrane is actually going to process sound before signals reach the receptors even, because it's just going to depend on which ones they activate. So if we unroll the cochlea, we'll see. At the base here, we get these short, stiff fibers. They react to those higher frequencies. While these at the apex, at the very end, it reacts to longer wavelengths. So they have more floppy fibers so that they can react to that specific wavelength. Okay, how does our equilibrium work? This is the last portion of this. Equilibrium is our balance. So our balance is maintained in response to the different movements of the head. From that inner ear, eyes, and stretch receptors in your muscles. They all help with balance. We have that vestibular apparatus. That is those semicircular canals in that vestibule. We have receptors in there. They monitor the status of equilibrium, so you know where you are in space. There's semicircular canals, the dynamic equilibrium. So here's those semicircular canals and those vestibule. The vestibular nerve goes to the vestibule and the semicircular canals because that's where we have our equilibrium. What does the macule do? That, of course, has sensory receptors, just like anywhere else. That's static equilibrium. So that's how we know where our head is in space, how we can maintain our posture. It responds to that linear acceleration forces that I mentioned before of being in a car that accelerates fastly. That's linear acceleration. Or being on a roller coaster. You know, on a roller coaster, some of those roller coasters first take off, they take off pretty quick, going straight forward. Being on one of those, that's linear acceleration forward before you start going different directions. The macule will not respond to rotation. So here is the macule. Just this purple portion through here. So what do we have inside that macule that lets us know where our head is in space? Just like in the cochlea, we have hair cells. They are stereocilia, known as kinocilia. We also have these otolith membrane that has otoliths or stones. These otoliths move across these hair cells depending on its changes in motion or inertia. The utricle macula are horizontal with vertical hairs. So they're going to respond in that horizontal plane, like tilting your head. Back and forth movements. The saccule macula, they're vertical. They have horizontal hairs. So just the opposite. They respond to up and down movements. So that saccule macula, when you go up or down an elevator, that's how you know you're going up or down. It's because of that saccule macula. Those hair cells synapse with the vestibular nerve, which is part of that vestibulocochlear nerve of cranial nerve 8. So here we see those hair cells, or that stereocilia, and that kinocilia. We have that otolithic membrane, which is kind of like a jelly, and those otoliths, which are those stones. So as our head moves in different directions, those stones push on that membrane that moves those hair cells that starts, which triggers an action potential, which sends that information on that vestibular portion of the vestibulocochlear nerve that allows us to know where we are in space. The macula. So those hair cells are going to release neurotransmitter that starts that action potential. So that otolithic membrane kind of causes a lag for a minute. So those hair cells takes a little longer to move before they cause that action potential to start. So as your head changes positions, those stereocilia move at the same rate as the head. But the tips that are embedded in that otolith, pulling of that is actually lagged. And that's what causes those hair cells to bend. When the hair cells bend, ion channels open, which causes depolarization, which causes that action potential to start. The bending of the hairs in the direction of the kinocilia will depolarize. So more neurotransmitters released. More nerve impulses get sent. When those hairs bend away, it hyperpolarizes, so not as many neurotransmitters are released. So not as many impulses are being sent. And that is how our brain knows that we have changed positions of our head. So here's the pictures that show exactly what we just said. Normally when our head is upright, there's no pull on those hair cells. So we get a steady stream of action potentials just knowing where our head is in space. But say you look down. By looking down, it causes those hair cells to bend. Bending of those hair cells triggers an action potential, which depolarizes, as it's called. So we get several action potentials sent right after another, after another, after another. It increases the amount of action potentials. What about when you look up at something? Well, when you look up, it hyperpolarizes. Those hair cells bend away from that kinocilium. It decreases the number of action potentials sent. So you can see they're less often. What about rotational acceleration? That is where our crista ampullaris comes into play. They are excited by acceleration and deceleration of the head. Rotational movement, like twirling, spinning in circles. The semicircular canals are located they're in all three planes. So that helps to pick up all types of rotational movement in that head. So we have supporting cells within the crista ampullaris and hair cells, just like other places. They have that gel-like that sits on top of the supporting cells in the hair cells, called the ampullary cupula. Here we have dendrites of that vestibular nerve that are at the base of those hair cells. So if we activate the receptors of that crista ampullaris, it responds to change in velocity of rotational head movement. So as you say no with your head, that crista ampullaris is responding. It causes the endolymph and the semicircular ducts to move in the opposite direction of the body's rotation, causing the hair cells to bend. So here we see that crista ampullaris. We see the ampullary cupula with all those hair cells down in here. So as we move our head to say yes or no, to say no, it causes these hair cells to bend. And as these hair cells bend, it's triggering action potentials down those vestibular fibers of the vestibulocochlear nerve. A vestibular nystagmus. It's how your eyes are connected to all of this. It's kind of interesting. So in those semicircular canals, we have impulses that are linked to reflexes of eye movement. And nystagmus is where your eyes kind of tick to one side. It's kind of a fun thing to do. I know I find weird things fun. But say you spin somebody around in a chair over and over and over and over again, and then you stop them and you look at their eyeballs. They're going to have this tick going the opposite direction of the rotation. That is the nystagmus. So a lot of times this is accompanied, you see it in patients with vertigo. You don't have to spin them in circles to get the eye tick to happen. Sometimes it happens on its own. Sometimes you just have to lay them down and they get that eye tick. But your central nervous system is trying to compensate. So it tries to jump in the rotation up towards the direction of rotation, but then it causes your eyes to tick back. So it goes the opposite direction, which is ends up being a jerky, rapid movement of the eyes. There's places where we can have problems with our equilibrium. And it can cause nausea, dizziness, and even loss of balance. And nystagmus, if we don't have any kind of rotational stimuli. Motion sickness is where those sensory inputs are mismatched. So visual input is different than their equilibrium. Like when you're on a boat, you don't always see that the boat is moving up and down, especially on a large ship. If you've ever been on a large cruise ship, you don't always feel that up and down movement going through the water, but your ears sense it, but the rest of your body does not. Your eyes do not match that up and down motion that your ears feel. That is what causes motion sickness. They can have excess elevation, pallor, so pale skin, rapid breathing, sweating. These people can take different anti-motion drugs because it depresses their vestibular input. Meclizine, scopolamine. Some of these drugs have fun names to say, or hard names to say, I should say. Dramamine, that is a form of meclizine. But these drugs help to depress your vestibular nerve from sending so many impulses about what is happening where you are at that allows you to not be car sick. People get car sick or get motion sickness in a car in many different places. So there's different people who suffer from this. There's many different drugs that can help them. This is the end of this chapter. Thank you for listening. Bye.